aerosols that require additional protection, that do not have any FDA approved intervention strategies. There is a risk of transmission. So those sorts of pathogens where we have to be very careful in how we deal with the pathogens, we work with them in the BSL-3 environment. So my lab does both cell culture based and animal based work with BSL-2 and BSL-3 pathogens. Next slide, Laura, please. So as I said, we have been doing this since, I don't even remember when we started. I've been doing virology work for as long as I remember, I think. We work with many acute viruses. What this means is that these viruses come in, they cause an infection, they cause disease, and they leave. So things like influenza, right now, what is happening in some ways, although it's still in the fuzzy area, is SARS-CoV-2. These are all short-term disease situations. So we work with a lot of those sorts of viruses, many different viruses. Many of these things you might not even have heard of them. They remain, thankfully so, exotic to some people, just because they are not so prevalent. We work with many viruses that cause encephalitis. We work with viruses that cause hemorrhagic fevers during late stages of infection. Many of these viruses are transmitted by mosquitoes, but many of them are also transmitted as aerosols. And as aerosols, they cause significant disease. So what is not included in this list, but what we have been spending most of our time with since early last year, is working on SARS-CoV-2. Again, because we have a BSL-3 lab, and working with SARS-CoV-2 requires BSL-3. In all of these cases, we say, how do the virus and the pathogen, how do the virus and the host communicate with one another? What is that language that they use to communicate with one another? Can you use that information to better understand what the virus actually needs to cause disease? And can you take those away? In other words, can you identify drug targets? Can you use FDA approved inhibitors, things that have already been approved for other indications? Can you use them to treat these conditions? That's called drug repurposing. We do a lot of that. We pay a lot of attention to inflammation, because in all of these cases, in every single one of these viruses, inflammation is an integral component that contributes to disease. It is the evil twin. So we look at inflammation in all of these things, and we have programs that look at vaccines in all of these cases. Next slide, please. And we do these in different model systems. So there is a pipeline, so to speak. You do a lot of work in cell culture first, and you identify things that make sense. Then you have to transition them into larger, more relevant models so that you can actually understand how a pathogen is controlled by a given therapeutic, how efficacious is your vaccine strategy. So we have small animal models for many of these things. These animal models also include exposure, treatment, vaccinations. All of these things are, kind of roped into these models. In addition to that, one cool thing that we have here is organ on chip platforms. So these are human-derived organs or representations of organs on chips. These are small glass chips or glass slides, and you have multicellular tissue environments reproduced in those glass slides. And so you can actually do some of these validation studies for these various candidates in a human microenvironment. And then moving on to the next slide. So we have all of these things that are in the pipeline. So this is going, all of these things are in various stages: cell culture, animal models, organs on chips. Right now, we have organs on chips for the brain and for the lung. We are looking to expand that to the heart, probably circulation and blood vessels very, very soon. Potentially the liver coming down the pike, but right now the big ones are the brain and the lung. We have multiple therapeutics that are going through the pipeline for many of these things. Many of them are FDA approved already. So we have a significant effort in drug repurposing. We also do a lot of work with peptides. These are small peptides, small amino acid, multi-amino acid chains, which have antiviral activity. But the cool thing about them is many of them are inherently immunomodulatory. So you can kind of get 2-for-1, buy one, get one free sort of a situation where you can have the same agent control the virus and also inflammation. We're also looking at biologics, many different kinds of antibodies, plenty of work ongoing with vaccines, many of them focusing on delivery platforms, DNA-based delivery platforms, protein-based delivery platforms, lipid-based platforms. And from a basic science perspective, we are also looking at what your immune system is seeing when a cell is infected by a pathogen. That is absolutely critical for you to be able to design a good vaccine and make sure that the vaccine is also presenting the same thing to your immune system. So those are all the kinds of things that are ongoing right now. So I think that is my last slide. With that, I will stop.
Wow, wonderful. Thank you so much. I forgot to mention in the beginning, if anyone has any questions for any of our faculty, please put it in the chat. I will grab them and if time permitting, ask them after each presentation. I have a question for you. I'm just curious, how long does it typically take for a study like yours to then be implemented into something that can then affect the public and actually be used?
Fantastic question. So that is where the drug repurposing comes in, because many of the initial IND safety, many of the things that the FDA would be looking for, we might already have answers for those. That a typical, start to finish pipeline can be anywhere from 5 years to about 7 years. To bring a vaccine to market, typically in a non-COVID situation, if all other things were equal, it might take anywhere from 7 to 10 years easy.
Thank you.
Thank you, everybody.
Well, thank you very much to Dr. Narayanan. We are now going to move on to our next speaker. We have Dr. Dieter Pfoser. He is the chair of the Department of Geography and Geoinformation Science.
So good afternoon, everybody. Microphone is working, I hope.
Mm-hmm.
Good. So I'm not only chair, I'm also a professor at this department. So geography and geoinformation science, what we are doing, at least what my research group and a couple of fellow faculties are doing, we are working on urban analytics. So we try to solve urban challenges based on the data sets that are available to us. And there's a lot of data available, and there are many, many challenges. Why urban is important? Well, that's because most of the people live in urban environments. And think of Washington, D.C., the Washington, D.C. metro area with its 2.4 million people. Now, the particular effort I'm going to talk here, and I called it Hack and Urban Mobility COVID Edition, is actually a hackathon project by a group of grad students who will be presenting this work today, actually in the afternoon, towards a panel of judges and their finalists, and hopefully it goes well and they're going to get a huge prize off of that. So I'm just posting also the link to the event in the chat. If anybody wants to join, it's open to the public, so you can just listen in. For example, Senator Warner is speaking at the event as well. So it's high profile. So next slide, please. So this particular project, hey, the animation works fantastic. So this particular project is about traffic. And as we all know, traffic is bad, or at least it used to be bad, right? So going to Washington, D.C. at the wrong hour of the day was really, we didn't like to do that because you're just stuck in traffic for hours on end sometimes. And COVID came along and suddenly everything is, traffic is not as bad anymore. Public transport essentially is hardly used at all. For example, the metro, 95% drop in passengers. Nobody uses the metro. Metro buses, nobody uses the buses anymore because just there's a fear of, crowding and getting infected. So next slide. So every day, literally now is like 5 o'clock in the morning, you can get on the road and it's fine. And a big reason is because we are all working from home, right? So we spend, like we do now, we spend our time behind Zoom, behind monitors on Zoom meetings, and we don't really have to go to work. And so this change, this fundamental change in work habits has really had a profound impact on transportation in the DC metro area because people don't need to commute anymore, at least for the time being. And one of our assumptions is, and that's next slide. So one of our assumptions is that some of those trends, because we have new work habits, are going to persist post-COVID. So the expectation is that our work habits post-COVID are not going to be the same as pre-COVID. And we, folks might be, that's too quick, folks might be able to work from home, for example, for 1-2 days a week. And so the big question is how it's going to affect traffic. There are other things like people might not use public transport anymore, because not using it for 1 year, you kind of are not in the habit of using public transport anymore, and you just might take your car, or you might use micromobility solutions or bikes and so on. So there are many things that will change when it comes to transit. And the question is, how can we kind of try it out? So how can we kind of reason about post-COVID scenarios in terms of transportation? Next. And our answer to that is really doing microsimulations. So what in this particular project is, we do large-scale agent-based modeling using a platform called MatSim, and using data about the population to parameterize the simulation. So essentially what we create is a digital twin of the DC metro area with its 2.4 million commuters every day, and trying out different mobility solutions. So as I mentioned it before, what would happen, for example, if folks would work from home, or a certain part of the population would be able to work from home 2 days a week, right? What would the impact of this be on transportation and on traffic in the DC metro area? Right now, nobody knows that, and we hope to have an answer for this one. Next slide. And a concrete example here is, everybody uses different transit or transportation solutions, and not everybody actually has access to a car. So there are areas, this is Fairfax County, so the areas in the county where there's a high percentage of people actually using the bus. And we saw in our data set that using the bus is directly related to income levels. So the lower your income, the more likely you're actually using public transport, or I have to say the bus metro usage is actually independent of income. So everybody uses the metro. But buses are really focused on more the low income population. So now budgets are tight at the county level, and there is talk about reducing the level of service for public transport, so cutting bus lines, thinning out the schedule of buses. And one thing you don't want to do is, you don't want to take away buses or the bus frequencies, or for parts of the population that heavily rely on that, right? So this is a concrete challenge that the county has. And the question is, so which buses can I reduce because they're not used, and which bus lines do I have to absolutely maintain at the level of service that I have right now? So and this is to guarantee transportation equity, as it's called, and to avoid transit or transportation deserts. So this is a concrete scenario that the county is looking at, and our simulation actually will have an answer for that for various post-COVID scenarios. Next one. So you might ask, so what? So one thing I mentioned, avoiding transit or transportation deserts, safeguarding vulnerable population. So folks that actually already struggle to make an income, to make a living, you cannot take away the bus. Otherwise, how would they get to work, right? So those are all very, very challenging problems that somebody like, the Fairfax County as a local authority has, because it's a 1.2 million population that lives in this county. So, and giving all that with budget constraints. So the money, the income, the revenue is taxes are gonna drop, and so the spending is gonna drop for the public sector and for local authorities. And the question is, where can you make cuts? And yeah, so those are all very interesting scenarios and questions, and hopefully the county will pick up on that and is interested in the work the students, so Dan Cheng, Shiji Fei, Tunajina Khan, Xi Yang Ran, and Meng Fei Jin are doing. So if you have the time, please join the team tonight at the presentations, and hopefully there is time, or there's a reason to celebrate later on, because they've won a prize. And that's it for me. Thank you very much.
Wonderful. I did see one question come through. The question is it possible to extrapolate this platform to figure out the dynamics of how a virus would spread in circulation? kind of think of the virus as the yellow line that takes the metro route, AKA blood vessels. Is that possible?
Yes and no. I mean, the problem is that, you know, it's people, it depends a lot, and there is, you know, other folks working on that. It depends a lot on the behavior of the population, right? It depends on whether people wear masks or not, whether they keep social distancing guidelines or not. So those are all factors that they play a much more important role than being on the same bus with somebody, right? So to give you an example, for example, my son, he's at UVA, and he was isolated because of, you know, some of his friends tested positive for COVID, and he has been around those friends for hours on end, and he never got COVID, right? And he was really close to them, so he never got infected because of that, and neither was any of his friends. So out of a group of 10 people, 2 were infected, but they didn't transmit the virus to anybody else. So it is much more complex than just being close to somebody for, you know, 15 minutes, but you can contract the virus. Of course, transportation plays a role, but it is not that simple. Let me put it that way.
Great, thank you so much. Really appreciate you letting us in on some of the things that you're currently working on. I'm going to move then to our next presenter. Just a quick mid-event announcement. I encourage you all to follow us on social media. Those of you who begin following us are going to be entered to win a prize. So if you would like to be entered into that, I encourage you to do so. We also talk a lot about our research on there, so it's a great way to keep connected with what's happening in the college. I'd like to now introduce Dr. Natalie Burls from the Atmospheric, Oceanic, and Earth Sciences Department.
Great. Thank you so much, Laura. It's a pleasure to be here and tell you a little bit about my research. Next slide, please. So the department I'm in is the Atmosphere, Ocean, and Earth Sciences Department. And so I'm going to talk a little bit about my research today, but really the students and faculty in our department are doing research on a wide range of topics. I've just sort of put it together here, a summary slide, but it spans both climate science and climate dynamics to geology and earth system science. And so a range of topics from the ocean's role in climate to paleontology and structural geology and tectonics. Next slide, please. So just to give you a little bit of background in terms of my path to Mason, I'm an oceanographer by training and a climate scientist. I did my PhD at the University of Cape Town, South Africa, so in the Southern Hemisphere where Coriolis force goes the other way. And at the time, I was also at times had the opportunity to go to sea and was lucky enough to visit the very southern tip of our planet, Antarctica. So here's a few photos from that voyage where really it's hard to believe that humans live on this planet. It's such a hostile environment, terribly cold, but the birds rule the earth down there. And so I love this photo. I decided to include this. It reminds me of Madagascar, the movie. And these penguins came up to us thinking, oh, another species, pretty friendly. Oh, wait, no, you're pretty tall. We're not going to come any closer. And then they carried on to check out the ship. But my love for climate science and research took me across hemispheres to the Northern Hemisphere, where Coriolis force now deflects everything to the right, and so it's incredibly confusing. But I've moved out of the sort of seagoing space and into the modeling space, and a lot of my research entails using climate models where we incorporate not only the ocean, but also the atmosphere, the land, and the sea ice and the land ice, and how they all interact to create our climate, the climate that we experience. And now I hang out instead with the GMU grad students. Next slide, please. So one of the key questions that my research focuses on is, if humans double the carbon dioxide content of the atmosphere from pre-industrial conditions, how much will the Earth warm? And so this is a problem and a question we can tackle with theory and models, but this is also we also have some observational constraints on this. We can look into Earth's history and see how it informs us, right? So my research uses climate models, but also data collected from past climate and past warm climates in Earth's history to understand the physics of this process. Next slide, please. So in this slide, I'm showing you climate records or reconstructions over the last 100 million years. And obviously, the further back we go, the more uncertain the data. But you can see here a very clear story. We have reconstructions of carbon dioxide and how that's evolved. And there's obviously uncertainty on these estimates, as I say, so that's the gray shading we have here on the plot. And then at the same time, we have estimates of the global surface temperature of the Earth. And so as we go back further and further in Earth's history, we can see that CO2 levels were higher, and that kind of goes hand in hand with higher global mean surface temperatures. And it's really in the last 5 million years that we've seen CO2 levels decline below 400 parts per million and to the sort of 200 parts per million, 280 parts per million that we had in pre-industrial times. Now, just because Earth's climate has been warm in the past doesn't mean that it's fine for us to be doing what we're doing right now. And what we're doing right now is shown in the far right of this plot. We're really applying an extreme perturbation in terms of elevations in CO2 concentrations, and we're making them rise very rapidly. And we have the three scenarios here. So in the more extreme case of the more extreme scenario of this sort of referred to often as business as usual, you can see we're taking CO2 concentrations back to values that they were last seen in the Eocene, for example, the early Eocene, where Earth's climate was radically different. And so I look at data from these, next slide, please. I look at data from these past climates, and I also try to simulate them with climate models and look at interesting things. For example, how different was the oceanic circulation during the Pliocene, which was the last time Earth's temperatures were, Earth's carbon dioxide was at 400 parts per million, but the continents were still pretty much in the same place, which makes it a fairly powerful analog for what we might expect to see in the future. Also looking at how the hydrological cycle, where it rains, where there's drought, has changed and how different it was during these past warm climates. Next slide, please. So in conclusion, I'd just like to say that we have a lot of research opportunities within our department, not only within the themes of future climate change and paleoclimates, but also the range of other topics that I mentioned earlier. And we have a lot of opportunities for interning through ASIP, for ASIP program, for example, during the summer for both the undergrads and high school students, as well. We have a very vibrant graduate programs in climate science, as well as earth system science, and plenty of opportunities to do research with our amazing faculty. So I'll leave it there.
Wonderful, thank you so much. I know that we have some folks on the line from your department, some students who are currently pursuing your academic programs, and those who just generally have an interest in climate science. So my one question for you, if there's a student who's looking for a faculty member to work alongside, or want to work with someone like you who's doing such interesting work, what is the process for identifying and approaching a faculty member to possibly then continue to work with them?
Yeah, that's a great question. And I think there's a number of different avenues. If you're an undergrad, especially in early stages of your undergrad, you could apply through ASIP, for example, and identify faculty members that you'd like to work with that would be doing a summer research. There's also obviously the research courses associated with our majors, as well as a variety of undergraduate classes, which you could use as a mechanism for meeting faculty. Otherwise, we have our pages, our faculty pages, and you can always feel free to directly reach out via email. Oscar is also another avenue.
Awesome, thank you so much. You posted those two publications that you recently, two studies that you recently done. So I'm actually probably going to grab those and put those on the ScienceConnect page after this event. So if anyone wants to read more about Dr. Burrells' research, you can go ahead and do that. So thank you so much.
Thanks.
So our last presenter today is Dr. Jennifer Salerno from the Environmental Science and Policy Program.
Thank you. Thanks for having me today, and thanks for coming. Next slide. So I'm in the Environmental Science and Policy Department, a wonderfully interdisciplinary department where I head up our lab, the Salerno Laboratory of Integrative Microbial Ecology, or SLIME, and that acronym was not an accident. We study environmental microbes, and most of the things we work with are pretty slimy. My lab is housed at the Potomac Environmental Research and Education Center, or PERIC, over in Woodbridge. And it's a fantastic new facility, as you can see depicted down in the lower picture there, right on the river. what allows us easy access to the environments we study. And there's a wonderful group of researchers over there, not only within my department, but also outside of the department that I collaborate with. And I encourage you to check out our website and see what everyone over there is up to and working on. The research interests and themes of my lab are particularly looking at the role of microorganisms in organism health and ecosystem function. So we study things like symbiotic interactions that then are impacted by environmental stressors, leading to what's called dysbiosis, or the breakdown of that symbiosis and disease. And we also look at how microorganisms can inform us about ecosystem health, because they are can so readily adapt to environmental changes and stressors. They serve as really good biological indicators. And so that's another area in which we work. Finally, being in the ESP department, I have a real interest in science and policy. And so I have worked in the federal government as a fellow in Congress and at the Department of State. And me, along with some of my other fellow alumni, have band together, and we've started to offer courses in science policy that'll start this summer. And we're working on developing a minor for undergraduates in science policy. So please reach out if you're interested in that. So students coming through my lab, I try to get them exposed to sort of these 4 areas of research, and that are really important to develop as a scientist and that are critical in our lab, but they also allow them to kind of figure out what they like, right? So I try to get them engaged in field work, sample collection, learning molecular techniques and microscopy in the lab that we use to study our microbes. We work with a lot of big data sets, so sequencing, so doing bioinformatics and multivariate statistical analysis. And then finally, outreach is a really important component as well. So getting them engaged, how to convey their research to different audiences, and also engage the community. So we do work on a lot of different things across that span, as I mentioned, but the one I'll touch on today is this disease we're studying, excuse me, called Stony coral tissue loss disease. And it's this disease that's infecting reef-building corals in Florida and across the Caribbean. So it first showed up in Florida in about 2014 and has since spread and is actively spreading across the Caribbean. So we're studying this sort of coral zoonotic epidemic during this pandemic. Luckily, we already had the samples because we can't travel much now to collect them. But it is a major concern because it impacts over 24 species of reef-building corals. And so down here is a picture of a coral that's been infected. So the brownish spots are where the healthy tissue is still remaining. And this is the active lesion, and it's completely eroded the tissue. And so it's got about a 50% mortality rate, and it's very lethal to these corals. And so it's of concern because those reefs are already impacted. So as of now, we are trying to identify the culprit. What is causing this disease? And we don't know. There's a group of national and international scientists working together on this. It was first observed in reefs offshore. So that's kind of interesting because it's away from population centers, but it is in areas where there is a lot of cruise ship activity. So it's thought that it is waterborne, and it may have been transported by passing cruise ships. Again, we don't know the pathogenic agent, whether it's a bacteria, a virus, a fungus, or if it's some sort of general stress response. And we're working to identify that. But there is some evidence that might be bacterial in nature, because out of resource managers trying to find ways to abate the disease, they've started to apply antibiotics, and it has caused cessation of the spread of the disease. So whether or not the bacteria is a secondary infection or the cause is being determined. So my research group, along with my colleague Esther Peters, are using both molecular approaches and microscopy to look inside the coral tissue and see what's going on. So we're looking at the bacteria that are present, who's there, where they are found within the coral tissue, as well as fungi that live in the skeleton. And we're looking at this endosymbionts that live in the corals called Symbiodinium or Symbiodiniaceae, and they are photosynthetic and they live in a beneficial symbiosis with the corals. But what's interesting is some of the initial results have looked at the coral tissue and found that this disease is almost eating the corals from the inside out, starting from where those Symbiodinium or Symbiodiniaceae are. So we're banding together to do that. And then through our coral reef ecology courses, we're also training students to do coral ID, and we're hoping to take them down to Roatan, where this disease just showed up in this fall, to do some surveys of healthy and diseased corals there. Next slide, please. So we like to think our research has a global reach, and we really approach our research through the lens of understanding the importance between the link between human health and ecosystem health. So we try to answer basic science questions, but at the same time develop environmental monitoring tools that can help with policy guidance and for resource managers. So we have a variety of state and federal funding for our research. And these are just some pictures of some of my grad students, undergrad and high school students that are performing field work. I have a student studying a disease in striped bass in the Chesapeake Bay, and then one of my other students doing molecular work in the lab. So as mentioned previously, ways that students can reach out, I think for all of the speakers today are through the ACIP programs. Oscar has several programs, the URSP or USRP, I always get that backwards. There's work study, and just reaching out to us and seeing what maybe volunteer or other paid opportunities might be available. So thank you for your attention.
Wonderful, thank you so much. I have a question. When you identify what is infecting these corals, is this a situation where the damage can be reversed, or is this a situation where we're just trying to stop the damage in its tracks and not have it go any further?
Yeah, great question. There's so much still yet to be learned. So in some cases, it seems that so first of all, some corals, like the branching corals, this doesn't impact them. Many other species it does. Some seem to be able to recover and fight off the disease. Others don't and die. And others, as I mentioned, if you apply an antibiotic paste, it can stop that disease spread across the colony, like the picture I showed, and they can recover. So we still don't know what the underpinnings of the genetics, the other environmental stressors, all the things that are interplaying into the spread of this disease. Something that recently came out is someone did an experiment and saw that it can infect Pacific corals, but it hasn't spread there yet. So we're trying to avoid that, but it is definitely a concerning situation.
Thank you. All right, well, I want to say a huge thank you again to Dr. Narayanan, Dr. Pfoser, Dr. Burrells, and Dr. Salerno. Really, really interesting topics that you all are currently looking at. Natasha, do you mind going to the next slide, please? So just, a recap of what we heard from a couple of our speakers today, as well as the dean. There are a lot of opportunities for our students to get involved in research here in the College of Science. Again, I will put this slide on our website. Some of this information is already there, but just to make it easier for you so you don't have to write down all of these long links. But there are ample opportunities for you to expand your own research portfolio and to get involved with what some of our faculty are already pursuing and already studying. So next slide, please. Just another reminder, I mentioned this before, I encourage you to visit primarily science.gmu.edu/research. It's where we post a lot of our research news. It's where you can learn more about our research centers and all things research in the College of Science. I also encourage you to follow us on any one of our social media accounts where we post news about our students, faculty, staff on a daily basis. And it's a great way to stay connected. Yes, Liam, I see your question. I will be putting this slide specifically on the science.gmu.edu/ScienceConnect page. So you'll have all of those links in one place so that you don't have to dig for that. So that'll be available there right after this event. I just have to get it up there. Wonderful. So the next part of our event, we're actually now going to go into our department breakout rooms. I've opened the rooms. If you have the most recent version of Zoom, you should now see the breakout room icon in your toolbar at the bottom. You should be able to, when you click on that, see all of the available breakout rooms and select which one you'd like to enter into. So for those of you who can do that, I invite you to select which breakout room you'd like to go to now. Don't forget, you are able to come back to the main room and go into another room if you should decide that you want to try to visit more than one. So for those of you who can do that, please do. If you don't have that functionality, sit tight just for a second, and then I will assign you to the room that you're interested in moving to.